Okay, so good. Hello and whoops, what's going on? Hello and so my mouse is a little funky. Hello and welcome back to our EEG class. So in the big scheme of things, we first had a rough and short tour through EEG. And then we dealt with the physics and the biological underpinnings of EEG, and we learned about this very basic model of how the EEG signal comes about based on the activity of pyramidal cell neurons in the cortex. And then over the last 2 or so sessions, we went over a number of what are called event-related potentials in the sensory domain first, and then also in the cognitive domain. And we learned about a number of phenomena that can be observed using EEG and that can be used to study the neurobiological basis of cognition. And now we want to turn more to the practical aspects of EEG data recording and analysis. And we start, of course, with the data recording aspect. So this is why the current session is called data acquisition. And we also have to talk about artifacts, so very common artifacts that are always seen in EEG, which are not of neural origin. So we have to find a way to deal with them so that they don't mask our signal of interest, which is the neural activity. So the topic for the current session and maybe the next, depending how fast things go, is EEG data acquisition and artifacts. Hello. There's something a little bit funky here, but anyway. So that's the outline. We first will talk about the basic technical thing that you need to record EEG, namely EEG amplifiers. Then we talk about the electrodes that are placed on the participant's scalp and how they are placed there. So that's electrodes and electrode placement. Then we have to talk about environmental electrical noise, because these systems that are used to acquire EEG data are quite sensitive because, of course, the biological signal is relatively small, and we have to make sure that we don't just record the electrical noise that is emitted by, for example, the computers that are used to provide stimuli to the participant. Then we talk about some aspects of electrode impedance minimization and why we do that. Then we talk about some aspects of recording and digitization, which are always good to know and which apply in any kind of digital data acquisition. And finally, we will talk about a number of physiological artifacts. So this shows essentially a very basic neurocognitive EEG lab. This is the EEG lab by the Center for Adaptive Rationality, so Ralf Hertwig's group at the MPI, which I had the possibility to build up a little bit before I then switched to Freie Universität. We have essentially the same system here, but we might not have as many systems as they have, and we might also not have a cabin. So what do we see? So here, this is actually back in this room here. We have some components of the EEG system. So we see some electrodes here together with the cables that go to the amplifier. And there are through two containers of electrode gel that we need. Actually, this is the gel, this is just the box. The setup then consists of the EEG stimulation room and the recording room, which is behind this door, and the recording setup. And you see that the computers are outside here. So you have computers here, you have monitors to see the recording. And inside the cabin, so this is an image from inside the cabin, you have a stimulation screen, and you have the amplifiers, you have a keyboard for button responses, and you can have, of course, a participant. And here you see how the whole setup that we are using here, which are active electrodes by Brain Products, looks if it's turned on. So here are the amplifiers, the electrodes are put here, connected to the amplifiers, and you can see that there are some lights on these electrodes which actually show you the impedance, so essentially the resistance, which you're trying to minimize by applying gel, and that's me here applying some gel. So that's how the whole thing looks, and of course you should, as for fMRI, you should take the opportunity to take part in EEG studies so that you make the experience how the whole thing feels from a participant perspective, and that you also learn something about it. Actually, the ARC EEG lab now with Bernhard Spitzer joining ARC will, I think, be put to more use than it has been over the last 2 years or so, and also with a PhD student who is one of your colleagues, Stefan Appelhof. We hope to record more EEG fairly soon. Of course, you can also record EEG not only at Max Planck, but also here. The only difference is that we don't have a cabin. They have a lot of money, so they build a cabin which shields to some degree the internal of the cabin from electrical activity. But you also see that we need some electrical devices in there, and then you can do all sorts of things to make sure that these don't interfere. And then science starts, and it's really a question whether you need such a cabin at all or not. But that's a scientific question. If you can afford it, why not buy it? Good. So I'm stuck here. So first thing to talk about, EEG amplifiers. So this is a magnification of the amplifiers that you just seen. So they are essentially just gray boxes, not too large, and usually one amplifier, the way the current setups is able to amplify the signal from 32 electrodes. Usually we will call it 64 electrodes, so we have 2 amplifiers. You see that this brain amp amplifier is actually MR compatible, so you can also use it for simultaneous EEG and fMRI recordings. These amplifiers, they are driven by battery packs, and here you see this battery looks from the outside like an amplifier, but it's just the battery to drive these amplifiers. So what do they do and how do they work? So when we talked about electrical potentials, we learned that the electric potential is essentially the amount of work that you have to do to move an electric charge from one point in space to another point in space. And depending on how the potential is distributed, this might be easy. So basically the charge wants to move there, or it can be hard, so you have to put in some effort and some work. But this then leads to the idea that the electrical potential is always a difference between 2 sides. Yeah? So you can only talk about electric potentials as a difference between 2 sides. And we discussed this question of having the infinity as a point of reference, but as we don't have that in real life, we always can measure potentials only with respect to some reference side. Now, this idea means that we need a couple of electrodes and differential amplifiers, and the electrodes use three kinds of electrodes. One type of electrodes are electrodes of interest or active electrodes, and these are the electrodes that are placed on the scalp locations of interest. So these are the electrodes that you later on use to evaluate EEG signals. A second electrode that you need is the so-called reference electrode, which is placed somewhere else on the scalp. Usually in post-processing, actually this reference electrode is replaced, for example, by the common average. And finally, there's a ground electrode, which is also placed somewhere else on the scalp. So for the brain active, the ActiCap that we use, the reference electrode is actually CZ, and the ground electrode is, where is it actually? Maybe we see it. I think it's also in the midline somewhere. So what is this about? Now, these differential amplifiers, they now amplify the difference, hence differential, between the following. The difference between the electrodes of interest and the ground electrode, so that's a difference. So they measure something, and at the electrodes of interest and at the ground electrode, and the difference between those is amplified so that we can digitize it. And the difference between the reference electrode and the ground electrode is also evaluated, and then the difference between these two differences is evaluated. So this is in kind of semi-mathematical form and a little bit easier to see. So the signal that is amplified is the difference between the signal at the active electrode minus the ground electrode minus the reference electrode minus the ground electrode. So for sure the activity of the ground electrode cancels out. So here you have minus ground electrode, and here you have minus ground electrode, so you have plus. So this sums to 0, and this leads to the fact that everything that the ground electrode picks up in terms of electrical activity, so fluctuations in the ambient electricity, is in principle filtered out or removed from the signal already at the recording, in the very early recording process. And this ambient electrical activity, which is detected by the ground circuit, is of course the same for all electrodes, and is here removed by only amplifying the difference between active electrode and ground electrode and reference electrode and ground electrode. Yeah. So this is why these amplifiers are called differential amplifiers, and then essentially these weak potentials that are picked up are amplified so that one can digitize them. So if you think that you have a certain bit range, and we will see that in a second, in which you can place over some continuous signal. Of course, you need to boost the signal to be able to actually discretize that with your given bit depth. Yeah, so we will not go into the electronics of amplifiers, how this actually works. That's interesting, but that's from our perspective here, this is kind of engineering. So for us, we content with the fact that the differential amplifier amplifies the difference between the active electrode and the reference electrodes, and hence we look at the signal as the difference between the active electrodes and the reference electrodes. What about these electrodes? What are they? So these are examples of electrodes. So these are Biosemi electrodes. So that's a company that does them. They are active electrodes, and here you see this little metal knob. That's actually the electrode. The rest is some circuit and some plastic shielding. Each of these electrodes has one cable that is then goes into this broad cable. Here you see a different kind of more old school electrode that is just a ring and has its own connecting cable, and here you have like a brain amp electrode setup, and this black one is actually the ground electrode, and the reference is, I think it's this blue one. And here you have another system, for example, for child EEG, where you have this net of electrodes which you just put on in one go, and then you can make very cute pictures. So what is an electrode? An electrode is just a conductive material attached to a wire. So simple as that. And as conductive material, what is usually used are metals with good conductive properties, and these are often silver. So this is often silver with a thin coating of silver chloride. So these are these AgCl electrodes, which are kind of the most common thing. They are electrically conductive, so whenever there's some current or potential fluctuations somewhere, they pick that up and propagate this, for example, to an EEG amplifier. The connection to the scalp surface is, of course, secured by means of a conductive gel. So this little gap between the electrode tip and the scalp is felt by a conductive gel. So that's just some chemical substance that is kind of gel-like and is conductive. That's a typical thing. The more fancy things that are being used are active electrodes. They have actually preamplifiers in the electrode. So it's not just a conductive metal thing, but it's actually already an active electronic circuit which boosts the signal. And this is helpful because it requires less gel and allows for shorter preparation times. And as we've seen, it also gives a direct feedback of how good the impedance is. Then there are other products, for example, by Geodesic, which are saline-based electrode nets. This is what we saw roughly with this baby there. So these you just put into water with some salt in it, and then you put the whole net onto the scalp. I've never used that myself, but the reviews are somewhat mixed on how good that is. And Brain Products also has developed some dry electrodes where you don't need any gel anymore, but you just plug them on to the scalp. So there's there's always some development. The conventional placement of electrodes on the scalp was according to what is called the extended 10-20 system. Which is, I'll show you that in a second, is a way that tells you in a standardized manner where you should put the electrodes on the scalp. This is interesting from a historical viewpoint. These days, everybody uses these electrode caps, so you basically just have to place the cap somewhat according to the standards, and then you just plug in the electrodes, and they are where they should be according to, for example, the extended 10-20 system. So what's this extended 10-20 system? We will see that in a second. So this shows the 10-20 system. It consisted or still consists, but is hardly used in this pure form, of 21 electrodes, which are placed at 10% latitude and 20% of longitude. These latitude and longitude notions, you might know them from geography. The idea is that you establish a coordinate system on the scalp, and then you measure these latitudes and longitudes with respect to this coordinate system. So of course you need to establish some references here, and the idea is that you have an equator passing through the Nasion, Indian, and the left and right periauricular points. So essentially what you look at is the Nasion, which is this place just above the nose, and you look for the inion of the participant, which is this little bump in the back of the head, and you establish the line from the nasion to the inion, and you measure how long that is. The same thing you do for these two periauricular points, which are just points very close to the ear. And then you have measured these distances on your participant, and then you divide the longitude between inion and nasion in steps of 10%. So this is what you see here. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So you measured, for example, I think, what is this usually? I think it's usually 36 or something. And then you divide this by 10. So every 3.6 centimeters, you then have a point for one of these circles. And then you, in addition, concentric to that, you introduce latitude lines again by dividing the distance between the periauricular points in steps of 10%, so going from the left one, so the left ear, to the right ear. And then you have this kind of coordinate system, and then the electrodes, which are named here, so for example O1, OZ, and O2, have a specific location within this system. And the 20% 10% of latitude and 20%. So the 20% is definitely these distances here. So here you see that there's always 20% longitude, and 10%, well, here you have 10 and 20% all the time for all of these electrodes. There are some exceptions. There are some exceptions, namely F3 and F4, which are halfway between Fz and F7 and F8. Do we see that? F3 and F4, yeah. So this is these are two exceptions, and P3 and P4, which are also halfway between Pz and T5 or T6. So they are not directly on these coordinate axes. So that's basically how people standardized the electrode placement before there were electrode caps. So there you had to measure the line from the nasion to the inion, and from the two periauricular points divided by 10, and then place these electrodes at the given point. So you could actually, I think what a lot of people used was to first measure that and then divide it, and then mark these things, for example, with some pen, and then put the electrodes there. This has then also, of course, led to be able to name electrodes and compare them between different labs or in different clinical settings. So the electrodes labels that are then used are first for the frontal pole, FP, for frontal F, C for central, so these ones, parietal for P, occipital and temporal. And the number and letter indicate the distance from the midline. So for example, if you look at the central electrodes, you go from CZ to C3, well, and then you're already at temporal. Can we see that a little bit clearer? Here, for example, PZ, so you go to P3 and then P7. So the larger the number, the greater the distance from the midline, and the numbers are according to the hemisphere. So the left hemisphere gets the odd numbers and the right hemisphere gets the even numbers. Yeah. So if somebody tells you we're looking at electrode C2 or C4 or something, then you know, okay, that should be on the right side and it should be fairly central. So maybe that's somewhere in the region of motor and somatosensory cortex, something like that. Yeah. Or somebody tells you we're looking at electrode 001, then you should know that's on the left side and it's over occipital cortices, so maybe more visually, and so on. Now, of course, this is the 1020 system with 21 electrodes, but these days there are people don't record only 21 electrodes, but usually 64. This is some more explanation, but I delivered that already. These days we use electrode caps. So here essentially one needs to make sure that CZ is placed, so the spot where the CZ electrode comes in is halfway between nasion and inion. So we still measure for each participant the nasion and inion distance, but then we don't divide it in steps of 10, but just divide it by 2. And at the exactly the half, the electrode cap hole for electrode CZ is placed, and then of course we also make sure that this is in the middle between the periauricular points, which you can, for example, test by tapping on it and ask the participants, but you can of course also measure that. Now then you have the electrode placed in principle according to the system, and then the electrode distances are so that they actually conform to the 1020 system. Now, that's interesting. There are the typical thing, of course, in when you acquire digital data is not to work with 21 or so electrodes, but to work with powers of 2. So the typical electrode setups are 32, and even more typical, it's actually 64. Of course, there are many more electrodes in between, but you have essentially you have this. You have a sheet that tells you which electrodes go somewhere, and of course they are also labeled, so you know where to plug in things. And with 128 you have even more electrodes in between. But it's still referred to as the 1020 system because these electrodes are essentially placed in the caps according to these old 1020 rules. But from a practical perspective, it's a lot easier. You just adjust the CZ electrode so that it's midline between nasion and inion and between periauricular point left and periauricular point right. And then you make sure that the cap sits nicely, and then you plug it in. There are different cap sizes, so some people have larger heads, some people have smaller heads, and this can be also measured in terms of the circumference of the head. Now, if you want to go even further in terms of standardizing and knowing about the location of the electrode, so here you essentially know it from the cap, but you can do more if you're really interested where the electrode sits with respect to a coordinate system of the participant's head. And this you can do with this 3D registration system, of which I currently forgot the name. I will see it on a different slide, but I also don't advertise this then that's also fine. So what happens here is the following. You first, of course, you need to express the location of the electrodes with respect to some coordinate system, and you first have to establish this coordinate system. Now, the coordinate system that is used by these 3D registration system is the following. You locate the pre or periauricular point left and right and the nasion. So this gives you this triangle in 3D space, and then the which is oriented, so you have some orientation of your coordinate system, and then the origin of the coordinate system is located at 40% of the ear-to-ear distance, which is also the best fitting sphere system. So you register these 2 points, and then you go 40% up from the middle and this is where the origin is. Now you have established a coordinate system, and then what you actually do to figure out where the electrodes are located in this coordinate system, you use this magic stick, which uses a Schallsystem, which uses Ultraschall, sonic. It is a sonic, supersonic is Überschall, so it's a sonic system. So this sends some tones, and these are reflected by this thing, which also registers then where the tip of the of this magic stick is. And the whole thing, what it delivers you is then a 3D localization of the electrodes with respect to the participant's head. So if for some reason a participant has a head like this, and you established where the origin is, and the participant has a bump here, and the one electrode sits here, then you know that this electrode was a little bit further out than all the other electrodes. The idea is that this can be helpful for source localization. I really haven't seen any studies that showed this in phantoms or something that this really helps. I haven't seen them. Maybe they exist. It's relatively reliable. So one thing that I did with this Cepris, that's the name, with this Cepris system was to actually put electrodes on a wooden head and measure this a couple of times, and it's fairly reproducible. So you can fairly good reproduce where the electrodes are, and then you have measured that, and then you have, in addition to the information that the electrode was actually located according to the extended 1020 system on your electrode, you have the physical representation of the electrode location with respect to a coordinate system that is established for each participant individually. Yeah? Good. Yeah. So a big question, that people discuss in the literature. You might know that there's currently also Frontiers research topic on the role of the reference electrode, because it's something that is not discussed much in the EEG literature. Everything is always measured with respect to a reference electrode, and then sometimes processed to be with respect to a different reference electrode. But the effects of this is something that is typically not discussed in the EEG results. And a big question is where the reference electrode should be placed. The thing is, ideally you would have an electrically neutral site, but then no side of the body and of a participant is really electrically neutral. This is what we've seen. What doesn't make too much sense is to use lateralized reference sites, because they then of course induce lateralization biases. What is more important from my perspective is that one always keeps in mind that the event-related potential waveforms that one sees depend on the reference site. Yeah, and this is just how it is. So you can, we did that 2 years ago or so in the programming project, which then people complained was too demanding, but nevertheless it was instructive. We computed event-related potentials from the data that you guys are also working on, if you want. We computed event-related potentials and then compared different re-referencing sites and looked at the event-related potentials. Then you see that it differs. It doesn't completely abolish things or something, but it differs. And so one should just be aware of that every event-related potential waveform that one looks at depends on the reference site, even if one does pre-processing. So the common reference scenario that is currently used is that the recording itself works with some arbitrary reference site. For example, in the Brain Vision ActiCaps, the FCZ electrode. And then offline, these data are re-referenced, so meaning that from each electrode, the must-read average, so the recording of must-read electrodes, so these are these bone protrusions behind the ears, or the average of all electrodes is subtracted. And then, of course, you can have more, can introduce more problems. So, for example, if you have a very noisy electrode and you subtract from each electrode the average, and this average is then contaminated by this noisy electrode, then all of these electrodes, after re-referencing, obtain some of this noise, and this is something you might not want, so you have to first exclude noisy electrodes. So yeah, it requires some thinking, and is something that one always should keep in mind, that the event-related potential always with respect to some reference site. And I think the most helpful thing is to, because it's so easy to offline re-reference stuff, to evaluate the data that you have with different reference sites. And then, for example, if you're looking at the difference between two event-related potential waveforms and two conditions, if you change the reference sites and the difference is always there, although it might sometimes go a little bit larger or a little bit smaller or whatever, then you feel better as if for example, the difference completely disappears if you use a different reference site, but that's unlikely. Good. So that was what I wanted to say about electrodes and electrode placement. Next topic, environmental electrical noise. So, of course, the voltage fluctuations that happen in the brain are really small, so they are in the order of microvolts, so 10 to the power of -6 volt. And to measure this in a digital manner, the EEG is amplified by a factor of 10,000. But this also means that environmental noise is that might be picked up is amplified and needs to be accounted for. The good thing about the line noise, so the alternating current line noise, which just comes out of the power sockets, is that it's very well described. It has a 50 Hertz frequency in Europe and a 60 Hertz frequency in North America. So you always in papers you see that they filtered out a 50 Hertz or 60 Hertz based on where they are. That's something that can be well dealt with. CRT video monitors, so the old ones that still had these electrode cannons that were shooting electrodes onto the screen, they also contribute to noise. LED monitors, so stuff that the monitors that are now being used, they are actually not well studied yet, I found, with respect to the artifacts they induce. I think there's one paper that I remember in Frontiers paper also that looked at that. Another noise source, of course, could be other stimulation devices. If you're not using visual stimulation, but some auditory or some other sensory stimulation, then you might have a device there that also creates some environmental noise. What can one do? Well, first thing is turn off any non-required equipment. What also sometimes helps is to change the location of the recording equipment. So you have to basically imagine that as that the electrical components that you use for EEG recording, they act as an antenna, and if you somehow change the positioning of this antenna, then you might not pick up line noise. So I remember recording EEG signals with the Biosemi system at Charité, and there we sometimes had the problem that we actually saw in the raw signal, we saw quite some 60 Hertz noise. And of course, one can filter that out using a filter that actually filters out, sorry, did I say 60? I mean 50 Hertz noise. And but what also helped is to move the whole system a little bit, and then get out of the region and space where this is picked up, preferably. And then, of course, one can, if one has a lot of money, use an electrical shielded chamber, which only uses direct current, so no alternating current, which might mask some of the alternating currents that we see in the brain. The question there is how much that really buys. So we have recorded actually an EEG set. That was when we set up the EEG system, and I felt I want to know whether this makes a difference, and to get started with the whole system and do some training also of PhD students and so on. We've recorded the same resting and task EEG inside the cabin and outside the cabin from the same participants, and we have all that data. But then a master student who was first interested in that from an engineering background was then not interested in that anymore, and the other PhD student left science, and so this data is lying around and hasn't been looked at all. So if somebody's interested in this data for a master thesis to figure out whether it makes a difference, then let me know and we can look at these data. Yeah. So whether you need a chamber or not, it's unclear. But if you have a chamber, then it makes sense to only actually have a direct current going in and not just put any cables in there, so that all the shielding essentially vanishes. Yeah. Good. Let's see what's this. I think we can do that relatively quickly. So the electrode impedance minimization is, of course, the most demanding thing during setting up an EEG. So you first measure the scalp of the participants, you put on the cap, then you plug in the electrodes, and then you fill this little spot between the electrode tip and the scalp with some gel, and you monitor the impedances at the same time, and you want to make sure that the impedances are as low as possible. And this, it's the most demanding thing, but you have feedback all the time. So this you see in these active electrodes, you have a little light that if it's red, then it's not good. If it's green, it's good. And you also have a digital representation of that, and you can actually save these impedances. So if you later then analyze the data and you see, oh, this electrode is very noisy, and then you can cross-check whether actually the impedance might have been a little bit large for this electrode. Yeah, so you usually start out like this, and you want to get there, and then you can start recording. We learned already that impedance is essentially the electrical resistance for alternating current or potentials, and that it, in contrast to resistance, it also has contributions from capacitance and inductance. It's measured in ohm, and you really want the impedance to be below 5 kilo ohm as a major step in EEG preparation to have good signals. What is the problem if you don't minimize that? These are two things. So first of all, the common mode rejection is less good, which means essentially that subtracting the ground electrode activity doesn't work, and what is known as skin potentials gets larger. Essentially, this whole thing means that the EEG noise is larger. So what is common mode rejection? It's the subtraction of environmental noise by means of differential amplification. And for some reason that is then also not detailed more in the logbook, common mode rejection gets worse with high impedance. The skin potentials are tonic electrical potentials between the skin surface and deep layers of the skin. And these change when the skin impedance changes, for example by sweat. And these create low frequency noise in EEG recording. And if you decrease the impedance of the skin, so by applying this gel, you also decrease the skin potential. So the fundamental finding is, if the electrode impedance is too high, your EEG is noisy. It's relatively clear, and you don't get good event-related potentials. The other question is why this is the case. And there are two explanations. One is this common mode rejection, which goes to a certain depth, but then also stops quite quickly, and the same for skin potentials. So they, it's not completely clear where these things come from, but these are explanations that you find in the literature. Good, so let's have a break now, and then after the break talk about recording and digitization, and then artifacts.